Welcome all. I am Dr. Dhivya R, Assistant Professor, Department of Zoology, Nirmala College for Women, Coimbatore. Today we are going to discuss on the topic basic concepts of development and the types of cell potency. Let's start with the topic. Developmental biology is a study of the processes by which organisms grow and then develop. The modern developmental biology studies the genetic control of cell growth, differentiation, and morphogenesis, which is a process that gives rise to tissues, organs, and anatomy. The development of an embryo from the fertilized egg is known as embryogenesis. All multicellular organisms arise by a slow process of progressive change called as development. Development is a process by which a multicellular organism arises initially from a single cell, that is called a zygote. The gradual developmental strategy is known as epigenesis, that is, the cell growth and division not only contribute to the size but to the shape and pattern as well as to the developing organism. So it involves five major overlapping processes. First one is growth, that is increase in size; cell division, that is increase in number; differentiation, that is diversification of cell type; fourth one is pattern formation, that is organization; fifth one is morphogenesis, that is generation of shapes and structure. Let's start with what is cell potency. Cell potency is a cell's ability to differentiate into other cell types. The more cell types a cell can differentiate into, the greater will be its potency. So the potency of a cell is an intrinsic property and is greater than or equal to its fate. The fate of a cell depends on its potency and its environment. For example, its contact with other cells in the embryo. Cell potency refers to the varying ability of a stem cell to differentiate into specialized cell types. So, cell with the greater potency can generate more cell types than those with lower potency. Next, we can see a zygote, which is considered as a totipotent cell. The zygote, from that fusion of an egg cell and a sperm cell, then begins cell division that are capable of forming the entire human body. So it is these cells that are totipotent, so called because their potential is total. Next, we can see the types of potency. First one is totipotency, then pluripotency, multipotency, oligopotency, and unipotency. First one is totipotency. Totipotency is the ability of a single cell to divide and produce all of the differentiated cells in an organism. That is, it can give rise to spores and also zygote. For example, you can see in the picture that is totipotent stem cell can give rise to either spore or it can give rise to zygote. Next is pluripotency. It refers to a stem cell that has the potential to differentiate into any of the three germ layers, that is endoderm, mesoderm, and ectoderm. Endoderm gives rise to the internal stomach lining, gastrointestinal tract lining, or the lungs, and mesoderm gives rise to muscles, bones, blood, urinogenital region, etc. And ectoderm gives rise to epidermal tissues and also the nervous system. Completely pluripotent cell can form every cell of the embryo proper. This ability to become any type of cell in the body is called pluripotent. So the difference between totipotent and pluripotent cell is only that totipotent cell can give to both placenta and the embryo, but a totipotent cell has a potential to divide until it creates an entire or complete organism. The pluripotent stem cell can divide into most or all cell type in an organism but cannot develop into an entire organism on their own. So that is the difference between a totipotent and a pluripotent cell. As the embryo grows, these pluripotent cells develop into specialized multipotent stem cells. Next one is multipotency. Describes progenitor cells which have the gene activation potential to differentiate into discrete cell types. For example, a multipotent blood stem cell and this cell type can differentiate itself into several types of blood cell like a lymphocyte, monocyte, neutrophil, etc. And the oligopotency is the ability of progenitor cells to differentiate into a few cell types. It is a degree of potency. For example, oligopotent stem cells are lymphoid or myeloid stem cells. So in this diagram, you can see that a totipotent embryonic stem cell can give rise to pluripotent embryonic stem cells, which in turn develops into endoderm lining, mesoderm lining, or ectoderm lining. And then the multipotent stem cells which can develop into lungs, pancreas, heart muscles, red blood cells, skin, neuron, etc. So the next one is unipotency. The unipotency cell is a concept that one stem cell has a capacity to differentiate into only one cell type. So it is currently unclear if the unipotent stem cells even exist, but unipotent stem cell has a capacity to differentiate into only one cell type. It can either become a circulatory system cell, or a cell of a nervous system, or a cell of a immune system. So we have reached the end of the class. Thank you. See you in the next video.